everyone. Welcome to today's webinar, Unleash Your Confidence with Advanced GC Solutions for Next Generation Chromatographers. I'm Megan Manzano, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by LCGC International and sponsored by ResTech. I would like to share a statement from our sponsor before we begin. Chromatography is what they do and who they are. They are an independent international team of employee owners serving analysts with unrivaled +1 service, applications, and expertise. From LC and GC columns to sample preparation, from standards to accessories, ResTech is your first and best choice. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player. The slides will advance automatically during the event. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. I would now like to introduce today's speakers. We are pleased to be joined today by Dr. Ramkumar Dandapani, Chris English, and Eduardo Morales. Ramkumar has more than 23 years of chromatography expertise, specializing in method development, validation, and troubleshooting. He has created regulatory compliant methods across multiple industries and now leads product management at ResTech, driving innovation and commercializing breakthrough solutions. Chris has led ResTech's innovation lab since 2004, overseeing new product testing, method development, and applications work. Previously, as an environmental chemist, he helped develop ResTech's volatile GC columns and holds a Bachelor of Science degree in Environmental Science from St. Michael's College. Eduardo has 25 years of experience at WEC Laboratories' Semi-Volatile Organics Department, specializing in method development and extraction optimization for emerging contaminants. He is skilled in GCMS systems, data review, special analyses, and supervising extraction teams. Thank you all so much for joining me today. Before we get started, we do have some polling questions for our audience. So I'm going to have the first one come up here, which says, which column do you currently use for your semi-volatiles analysis? And I'll give everybody a couple of seconds to answer this one. All right, great. We'll move on to our next polling question for our audience, which says, what are the current challenges with semi-volatile analysis that your lab is encountering? The options here are solvent ban and lower volume extraction, column lifetime, peak shape for challenging analytes, sensitivity and lower level detection, or other, which we would ask that you please specify what that is. We'll give everybody a couple seconds to answer this one as well. All right, great. And we'll move on to our last polling question before we begin our presentation, which says, which detection mode do you primarily use for semi-volatile analysis? Being GCMS, GCMS, or other, which we do ask you to please specify, and we'll give you a couple of seconds to answer this one as well. All right, great. Thank you so much to our audience for your participation here. With that, I'll hand it over to Ramkumar to get our presentation started.
Thank you so much, Megan. Again, I'm so pleased to unleash lab's performance by enhancing detection, lowering downtime, as well as improving more discovery within the lab using RMX GC column. Thank you very much for your time. Let's talk about the high-level overview of this presentation, right? So the first thing I wanted to focus here is the current challenge that the market is facing, and then how this new technology, Trimax technology that comes with RMX, helps solve this problem. And then I'll transition this presentation to Chris English, who will talk about more real-life examples and applications. And then we'll transition it to Eduardo Morales, who's an expert in environmental testing. All right, without further ado, let's get started. All right, talking about challenges in market, ResTech takes great pride in understanding how close the market is and how we wanted to help our customers by being in the front of the problem and then helping people with a solution. On that note, we did extensive understanding of customers to see what problem they are currently facing. One of the biggest problems that we came across is this methylene chloride ban. As you know, methylene chloride is a chlorinated solvent, and because of the nature of the chemicals in it, it actually is hazardous to the environment as well as to human being when they are exposed to this solvent. So now it doesn't stop there. The methylene chloride ban that's imposed has a huge impact on EPA methods. If you look at EPA methods, there are a lot of methods that utilizes methylene chloride as an extracting solvent. It has great efficiency in extracting analytes from water-based sample and different matrices. However, because of its impact on people's health as well as on the environment, the ban has been imposed. Now, the ban actually cascades further as well, right? So the disposal cost of methylene chloride goes really high. And then the exposure limit has also been reduced for people who are handling methylene chloride. And storage also becomes an important issue. So now labs are looking at ways where they can actually reduce the consumption of methylene chloride or go for an alternate method of extraction or lower volumes of different solvents. So that's one of the primary challenges that this industry faces. The next big one is I would also call this as an opportunity rather than a challenge is method consolidation. 10 to 15 years ago, when you look at methods, there were multiple methods for testing. Now people are trying to actually consolidate those methods so that with one or two tests, they can actually release their samples. A good example is combining semi-volatiles with another class of compounds, for example, a PAH or PCB, or combining PAH with another method, maybe a PFOS, right? So this type of consolidation actually is being seen across the globe and which helps people to actually improve their throughput in the lab. The first two challenges has actually pushed customers to lower the detection limits, right? So they want to see lower levels of analytes in their sample. They take help of the instrument as well as they need help from a column which can actually achieve those lower level detection. In addition to that, the inlet and the liners also play an important role. So as a bundle, right? So when you look at an optimized inlet and then the right column that can hit that sensitivity level along with your instrument can actually help overcome these challenges. Last but not least is also the lifetime of a column. Again, it's a very debatable topic when you talk about lifetime. It's matrix dependent. However, extending the lifetime and also having a column that's resilient to multiple matrix has a huge impact on the lifetime of the column and also going high throughput and saving cost per sample. On that note, let me introduce you to the latest technology and innovation that Restec is bringing here. And prior to talking about this innovation, I also wanted to talk about how it was viewed maybe 15, 20 years ago versus 5 years ago. So let's talk about the legacy deactivation in GC columns. If you look at legacy deactivation, they used industry chemicals that are not intended for GC purpose. Now, if those chemicals are not intended for GC, what happens is they might be slightly bigger in size, so has a lot of steric hindrance. So they might not provide the optimal coverage of the surface. So this actually results in surface imperfection and activity. So you would see peak tailing, adsorption of the analytes, and then that could actually result in a low throughput method. Now, considering these methods were analyzed 15, 20 years ago, all those methods were looked at in ppm levels, so higher concentration. At that level of concentration, those imperfections did not matter. Now let's move one step ahead and look at the timeline. We are talking about non-Trimax deactivation. So the depositing reagents used in the manufacturing process actually could create byproducts that can stay in the column. Now, the deposition of these reagents within the column, again, this might be minute, but actually they create residuals inside the column that can actually catalyze your stationary phase itself. And also, there are spots for adsorption of analyte. Here as well, the results is bad peak shape, column variability from one column to another, and then sensitivity issues, and then peak shape issues. Again, at this stage, we were talking about more of a higher PPB level detection. Now, as we spoke about in the previous slide, people are trying to overcome a huge challenge of this methylene chloride ban, right? So now people are going for really low level detection, right? We're talking about picograms on column or zero point 0.5 to 1 ppb levels. So again, we can rely on the MSMS instrument, but it's equally important to have a technology that can actually give you confidence in your lower level detection. So the Trimax deactivation is an innovation that uses novel reagents which are intentionally designed for GC. When I say intentionally designed for GC, you can very well understand that GC analytes are volatile. Now, these optimal reagents and process actually, because of the nature of the chemical and how it's designed, leaves minimal residue onto the column. Now you get a pristine surface that's synergistic and chemically yields a great order and dense cross linkage. So, in addition to having minimal residue on the column, now you also have an intact polymer throughout, which is synergistic. So, it builds resilience within the column in addition to enhancing that low level sensitivity. You get symmetric peaks, enhanced column lifetime because of the synergistic chemistry, and reduced column variability between column to column, and also upon continuous use of your matrix onto the column. So here is a zoom of what I predicted about the Trimax technology. Let's look at the top image where we have a non-RMX deactivation, and it's a non-Trimax deactivation basically. So now when you look at here, you might see those yellow spots which are residuals. Now, if you have inactive compounds like nonpolar aromatics, then there is no big issue there. While when you look at active compounds, we are talking about acids, bases, alcohols, esters, which can all form secondary interactions if there are any active spots present in the column. As you could see here, the top chromatogram, it can have some type of bias to the analyte because of the active spots. While with the Trimax technology and RMX, it is a seamless deactivation. It's a three-dimensional array of bonding, cross-linking, and deactivation, which has almost no active spots on the surface. So the column sees actives and inactives in a very similar way. So it's very unbiased to acids, bases, and neutrals. You see sharper peaks for all these, especially at lower level. This is of greater benefit. So here is our RMX GC column with Trimax deactivation. As I mentioned in the previous slide, Trimax deactivation is a three-dimensional array of bonding, cross-linking, and deactivation that maximizes trace level sensitivity and inertness. And it also yields a very uniform stationary phase, which extends the lifetime and resilience of the column. As you could see, can see a gradient of that bonding, cross-linking, and deactivation layer. So it's unified and it's very useful for lower level detection. So these are all the engineering design of this innovation. Let's look at what are the benefits you would get from this design. You get enhanced detection, and then high level, it lowers your downtime because there is resilience built in the column, and this helps you do more research, more discovery out of your GC column. Now, if we break that down further, because of the Trimax technology, we're able to look at analyzing wide range of analytes. We are talking about unbiased results for acids, bases, and neutral compounds with confidence. And then you'll achieve lower level calibration. We are talking about picogram level sensitivity and then single digit ppbs. And then this column helps you achieve those limits. And then it also maximizes your productivity by extending method performance and giving you longer lasting calibration over extended period of time. Now, this column is also backed by Restex quality. As you could see here, the left-hand side, we have 4 different batches of polymer. And as you could see, the polymers are very intact. We have put this column to extreme test, and you could see that the fingerprinting of all the 4 batches in terms of retention. The column is very intact and the process is intact. So we are able to provide you that reproducibility column to column and batch to batch. On the right hand side, you can see a lot of challenging analyte. I'm talking about weak acids, weak bases, aromatics, nonpolars, and also alcohols in this mixture. So we have really challenging analytes that we make sure that the column quality is maintained. And also when you look at the test mix, we are testing these columns at nanogram level on an FID. So when you get this column, you can be very confident that you can reach those picogram limits on your triple quad mass specs. So here is yet another example of how Restec backs this column with our thorough quality. You could see that 120 columns have been evaluated, a tight window of efficiency for those 120 columns. Again, this is an explanation that can build your confidence that every time you get the column, you can expect similar results. On the right side, as you could see, there are multiple analytes we put into test, as I explained in the previous slide. And it's not just one parameter we look at. We look at various parameters, including the peak heights, pars, the symmetry of the peak for acids, bases, neutral compounds, resolution, all those, so that you can have that confidence that when you open the column, it's right for your method and it serves your sensitivity requirement. Here is another example where we are comparing an Integra guard with a non-Integra guard, right? So the intent of an Integra guard is to extend lifetime of your column, meaning if you have dirty samples, then you can trim the sacrificial part guard and then not compromise on the resolution. So what we are trying to show here is when you have an Integra guard and without an Integra guard, the peak shape and sensitivity remains intact. Thanks to Trimax technology, which provides you that seamless deactivation throughout the analytical column as well as through the guard, where you could benefit the peak shape and sensitivity from this technology, even with an Integra guard attached to the column. So now let's talk about various formats that are available in RMX. Obviously, the analytical format is available for lower level sensitivity requirements. And in addition to that, we also have various different formats. As you could see, there is an Integra Guard version. So this is specially intended for people who have really dirty samples and they wanted to trim their column multiple times and reuse those without actually shifting the retention time. So integral guard is a seamless union of the guard column with Trimax deactivation to your analytical column. So you don't need a secondary part to actually integrate it. It's a leak-free setup that's most suitable for mass spec-based analysis. The other two options, which I'm super excited to share to you, are the Integra transfer line and also the dual Integra guard. So when you take a mass spec setup, the transfer line is usually kept at the highest temperature, right? So now when you put in a column with stationary phase in there, there is opportunity that the stationary phase can actually spit onto the mass spec source. Now, with an Integra transfer line, you have a deactivated Trimax tubing there without the stationary phase. What that encourages is lower noise level and better boost of your sensitivity. And then now let's talk about something even better. You have a dual Integra Guard version, which has the Integra Guard in the front end as well as the Integra transfer line in the back end. Now you get the benefit of an Integra Guard as well as an Integra transfer line in this format. So based on your specific needs, you can choose one of these and then get the best out of these dimensions. On that note, I will transition these to Chris English, who will be talking to you more about real world applications. Chris, over to you.
All right. Thanks, Ram. So in this particular part of our presentation, we're going to talk about 8270 method performance, semi-volatiles, low-level performance. We're going to evaluate single MS versus MSMS and do a comparison. So here are the different applications that we'll take a look at. There are the GCMSMS applications, and then there are two MS applications at the bottom that we'll discuss. And all of these will highlight the advantages of using this column over a standard column format. So again, the kind of thing that we're looking for is reducing the amount of methylene chloride usage. Currently with the 8270 method, when we do extractions, we're extracting up to 180 mils using a liter of water, which would be our sample. We can drop that down tenfold or more if we can see compounds at lower levels on column. And that's kind of the goal for this particular column stationary phase, is to be able to see active analytes at extremely low levels, and that's picogram levels on column. So we're in this center slide, we're going from a single ion monitoring type chromatogram, which is still going to pick up quite a bit of noise, to MSMS, where we're looking at the fragmentation of an initial fragment or spectra, and then doing a secondary fragmentation to get an extremely low level or sensitivity for that column. So this is a transfer from GCMSMS to MSMS. Again, the labs are pushing for higher throughput, and that's by combining different methods as well. So we're combining 8270 with pesticides and PCBs and other things as well. So those laboratories are going to be able to do a whole lot more with less analytical runs and less calibration runs. So here where we're looking at the deactivation and calibration, the ideal is going to give us a linear calibration. In this case, we're looking at correlation coefficients of 0.995 or better. Or if we're looking at relative response factors for relative standard deviation, we're targeting below 20%. And we'll show different graphs where our column outperforms traditional and competitor columns. If we look at a response factor for a parabolic type curve, which is here, even if you go with a quadratic calibration, you're still losing accuracy. And every analyst knows that if you can do a linear calibration, then that's the gold standard for calibrating the instrument and having accurate results throughout the course of the run. Obviously, with a traditional column, that's not always possible. And then again, with lower concentration and also having a linear response, we all know we can get a good linear calibration if we are running at higher concentrations. If we're calibrating from 5 nanograms to 200 nanograms, chances are good we're going to have a good linear calibration for all of our compounds. But let's say we're going down to picogram levels, single or double digit picogram levels. That's where this lower concentration, it's much harder to get a linear response. So in this evaluation, we were looking at method 8270E, calibrating from 0.5 to 5,000 ppb, and evaluating the compounds throughout that list, and then doing the same calibration for traditional columns and competitor type columns. What we're going to see is what we showed you with the ideal type calibration versus a competitor calibration. Again, this is for very low levels, all the way down to 0.5. So if we look at our percent RSD for a calibration curve, in this graph, you can tell that we are below 10%. So the below 10% is better than 80% of the compounds. And so this is a really good graphical representation of how solid this column is for very low levels. The correlation coefficient is even more indicative of the performance of this column with the calibration curve, because we're better than 90% for 0.995 correlation coefficient. And this is one of my favorite graphs here, and that's a low calibration point. Now, environmental labs do not run a low calibration point and calculate it against the calibration curve because the results don't always look very good, and they generally do a midpoint run for a continuing check standard or continuing calibration verification standard. In this case, we ran this low calibration point simply to show the accuracy and the linearity of the curve. If your curve is not linear, you're not going to get good recoveries down at the low end. And that's exactly what we see with other columns. And again, we're north of 80% of these compounds are within 70 to 130, which is an acceptable range for a low calibration point. Now, if we're going to go with the continuing check standard, which would be at the midpoint, so we're going to start right, or we're going to try it at 50 parts per billion. We have really good recovery when we're calculating this against the curve. And again, this is above 70%, between 70 and 130%. And you can clearly see that it does outperform the other columns. And then if we look at replications, so if we do replicate analysis, we also see that we have really good run-to-run reproducibility when we run a 50 ppb check standard repeatedly and calculate it off the calibration curve. So all of those show positive results for this column and show that this column does really well at low levels for active compounds. So, one of the things that, we've had questions is, well, maybe you acidified the column. And if you acidified the column, then your acids will look really good and your bases will suffer. But, overall, the column might actually, on average, look better than everyone else's. And that's just, that's not the case. The more neutral you can make this column, the better it will perform for both acids and bases. And that's what we see here. And, neutrals, there may be a slight edge on neutrals. And I've spent time trying to figure out, well, why would neutrals look any different? They shouldn't. They should all look identical. But Ram showed that one slide with efficiency, where our columns are consistently at 4,500 plates per meter. If you have better efficiency, you have narrower peak widths, you have better signal to noise, and you have more reproducible peaks, which means that even the neutrals are going to look slightly better. And so that's kind of the explanation. That's my explanation for why our neutrals even look better on this column. So if we're looking at method detection of quantitation, these are some other ways that we can evaluate the low end and show that this column outperforms other columns out there. And here's an example of method detection limit in parts per billion. Again, if we're running this, we're averaging about 1 part per billion for a method detection limit. Other columns are going to come in around 2 parts per billion. Minimums 0.05 versus point. Here's where it kind of shines, where we behave well or are active, react with the surface or react with the injection port or other places, we see a pretty dramatic difference. And in that case, we have an MDL of 50 in compounds, whereas our highest MDL is 14.47. Again, for the lower limit of quantitation, which is what we'd set our curve at, we also see differences here, 14 versus 20. So we are seeing differences over for all of the comparisons that we've done. So if we do an analysis with 150 different semi-volatiles by MSMS, and then we're running that from 0.1 to 10 picograms on column. So this was another kind of stringent test that we ran, and we have some examples of what that looks like at the very low end. Here's a striking example for pyridine. Pyridine on some columns is difficult even at 50 picograms to analyze. And we did, you do need to optimize pyridine because it comes out very early in the chromatogram, right next to N-nitrosodimethylamine and 1,4-dioxane interfere with methylene chloride. So optimized injection port conditions is required to get a good pyridine peak shape. And then once you've optimized that peak shape, this is what you'd expect to see on the RMX column. So excellent peak shape at extremely low levels. Again, 2,4-dinitrophenol is a very difficult compound. And then all the way down to the benzo-BK, which, this separation is quite adequate at these extremely low levels. All right, so one of our collaborators, Shimatsu, agreed to try out the RMX column. Now they have a new instrument, the TQ8050, which is easily able to see down to picogram levels of sensitivity. So this was a really good opportunity for us to try out the RMX column on one of their top-of-the-line instruments. If we take a look at the chromatogram here, we have a really good peak shape all the way throughout, and you can see all the way to the PAHs towards the end of the chromatogram that we have really good signal to noise and really good peak shape. And that speaks to the instrument as well as the column. So that instrument has a hot enough transfer line that we get very efficient transfer of the PAHs to the detector through the source and then to the detector. If we take a look at the low point from their calibration, they went down to 5 and all the way down from 5 and 10 ppb or picograms on column. And you'll look at their correlation coefficient is 0.993 or better for all of the compounds. And then you'll look at their detection limit. Their instrument detection limits were extremely low as well. So this is just a really good example of the RMX column and how well it performs just right out of the box. So MSMS analyses that we have, we can take a look at low-level single-ion monitoring by GCMS and regular GCMS applications. And again, really, the name of the game is to reduce the methylene chloride, so to inject lower concentrations onto the column which requires us to extract less. And if we're able to switch from GCMS to MSMS, we can save a lot of money on the extraction side of things. We can also combine methods with MSMS because we do reduce a lot of the background noise using that type of instrumentation. So in this case, this is the GCMS, and this is a 1 nanogram on column, which we easily have the sensitivity necessary for this analysis going down to 1 nanogram. That's certainly not a problem, especially on a standard, newer GCMS system. Single line monitoring, again, we have plenty of signal down at 200 picograms on column or 0.2 nanograms on column. And down here in this chromatogram. So either way, we could do a regular analysis with a GCMS, or we could do the single line monitoring if your clients allow it. In this case, we also looked at the Chinese method, the HJ834 method, and evaluated all of the criteria. And we're completely acceptable with the criteria. And I think the thing to also remember is with these difficult samples, this column will be able to pass the criteria for a longer period of time because the surface does not get contaminated as easily and become reactive. And a lot of that has to do with that Trimax deactivation where the entire surface has been covered. So one thing that we have, this is an actual chromatogram, and we have the method conditions here. So we ran the method, and it's about a 1 hour run. We loaded the compounds into ProEasyGC, optimized the conditions, and we're able to come out with a 36-minute run that still meets all of the criteria of the method. And so by using EasyGC, and you can optimize your particular method and speed up your runtime. And in EasyGC, we have all of the libraries, the major libraries, which would be the pesticides and the semi-volatiles. We also have fluorotellumer alcohols now loaded in, and you can either pick GCMS or GC to resolve the compounds because those compounds can be resolved by ion. And if you ask EasyGC to solve using GCMS, it will resolve them. It'll target like benzo-BK to get those resolved by ion. If you just pick GC, it's going to try to resolve all of the compounds. For liner deactivation, we have the Topaz liner, which is our Topaz deactivation. And it's a silanized glass. And we highly recommend it if you are going to try to go to extremely low levels. You need a liner that can also go to low levels. And our wool is in situ deactivated. That means that we have a vapor deposition process where it's deactivated after it's been put in the liner, as opposed to grabbing some wool and putting it in the liner, which then breaks the ends of the wool and exposes silanol sites, which are going to be extremely active. So our liners are designed for this kind of analysis. And again, I would recommend the precision liner for a splitless or for a split injection, and then a single goose with wool for a splitless injection. So in summary, our goal is to reduce the cost for the laboratory. And if we can reduce the methylene chloride and reduce the shipping of 1 liter samples around, going to reduce the cost. And then also by combining methods, this allows many more analyses to be done per run on this column.
Thank you, Chris, and hello, everyone. And I had the opportunity to try the new RMX column for 6 months now, and I would like to share my thoughts and how it worked for me. But first, I would like to share a little bit of who we are. So we are a full-service environmental lab specializing in drinking water, but we're also proficient in other fields of testing, such as wastewater, pharmaceuticals, food, and when I mean full service, I do mean full service. We are pretty much a one-stop shop. We provide extensive field of services across different parameters, with a broad analytical coverage across different matrices, from drinking water to microbiology to field testing and a few other more. We serve different clients from numerous sectors and industries, from landfills to municipalities and private water utilities. And our reach goes beyond just California. We work with other countries across the globe. And from my experience, in my opinion at least, the reason for, I think, our success is that we stay at the forefront of technology and how the industry changes. And one of these changes is the upcoming methane chloride ban. On April of 2024, EPA determined that DCM presented a risk to human health and issued a rule that under the Toxic Substance Control Act, which prohibits most commercial and consumer use of methane chloride. So while the new rule allows continued use of DCM, it will require more control and documentation if it's being used. And it makes sense, right? Because there's well-documented links to cancer and organ damage with methane chloride. So it makes sense for the labs, or for at least for EPA, to lower the exposure limits just to make everybody work in a safer work environment. So why does that matter to our lab? Why is that of concern? One, it's going to impact our operations. A lot of the methods that use DCM, it's used as a primary solvent. So a lot of legacy methods like 8270 and 525 uses DCM as a primary solvent. So it's going to force labs to reevaluate our workflows and reevaluate our DCM usage. It's also going to impact compliance. There's going to be a mandatory exposure monitoring. It's going to lower the monitoring from 25 to 2 ppm on an 8-hour period. So it's also going to assure that our hazardous communication plans and training and SOPs are updated, and the inventory of what's stored in the lab, it's also going to be well documented. And the bottom line is going to affect the financial impact. The DCM ban increases, it's going to have essentially a shortage. It's going to be produced less. So the lack of production is going to increase the cost, and it's going to go up throughout the years. And I see that already. The cost of DCM, it is going up. Also, disposal. Getting rid of methane chloride, we don't use 100% of the methane chloride. So we have to have somebody to come pick it up and treat it and dispose it properly. That's also going to increase in cost. And so the solution is to reduce it, right? And it makes sense to reduce it. So it makes sense to use less. It's going to improve the worker health and safety. It's going to have a better working environments at the lab. There's productivity gains. Instead of having to utilize 200, 1,000 mLs of methane chloride, you will be using fewer amounts. Like you'll be using 10 mLs. There's environmental benefits. Having less methane chloride in the lab or on the lab, it's going to produce less fumes, less methane chloride up in the air, and it's going to align better with your corporate sustainability goals. Cost savings, like I mentioned in the previous slide, because methane chloride is going to go up. Purchasing less methane chloride is going to be decreased. Decrease in purchasing, cost saving in purchasing, and cost saving in disposal. In terms of the footprint of the methane chloride around the lab, instead of having 1,000 liters of methane chloride, you would be purchasing 100 liters. And also lowering the hazardous waste classification would improve the lab situation. So what are the method challenges? Nothing's going to come free, right? One of the issues is that EPA validates with a specific solvent to sample ratios. So using less methylene chloride, it's going to equal to a lower extraction efficiency. We can't just decrease the amount of methane chloride while maintaining the same amount of sample. A practical solution would be to scale down. So if you scale down by 25% of methane chloride, you have to scale down the 25% of the sample as well. This maintains the extraction kinetics and the phase ratio, and avoids you having to use a different solvent that's not allowed by the method, or it doesn't work as well as methane chloride. So it's more practical to keep methane chloride as part of the procedure. But the requirement of this, if this is done, is that we're going to need more sensitivity. And because we're extracting less with less methylene chloride, the sample volume will be reduced, and then the analyte mass will be reduced as well. So you're going to need a lot more sensitivity. So the solution for this, it's using a more efficient tool or a more efficient technology, something that lets you see a lot lower than what you need to see, than what you're looking at right now. And this would be the Restec RMX column. I tested this column for a few features: stability, sensitivity, resolution, efficiency, and robustness. Stability, I needed for this column to have low column bleed and reproducible. I wanted my shot from one shot to another to be the same, and while also having a low thermal bleed. Sensitivity, I'll need to see this, make sure the peaks I see are good enough for me to report. I need to see my reporting limit. For resolution, I need to have the ability to resolve a few isomeric peaks that behave pretty much identical, and even some non-isomeric peaks that collude with each other. And efficiency, I need to be able to do my run in a timely manner, right? I can't run for 2 hours. I need this analysis to be done in 10, 20 minutes. And robustness, and how long will it last? Having a column that's great and gives me great sensitivity and great resolution, but only lasts a month, is not efficient for me. They're expensive, and it's troublesome getting a column out of the instrument and back in the instrument. It just doesn't work that way for productivity. And it tested this column on a couple of methods. On 525 by GCMS, which is a drinking water method, and 8270 by GCMS-MS, which is a triple quad method. These two methods they have overlapping analytes and run through a different similar compounds class, from pesticides to pHs to phthalates. So it's a little bit of everything, which is a good test for this column, because there's a lot of different kinds of different types of compounds. So the first thing I tested for is thermal bleed. So if you look at redirect your view to the end of the analysis, at the high end of when the temperature of the oven is really, really high, traditional column tends to give me a lot of thermal bleed, which lowers my reporting limit, lowers my sensitivity. It creates more of a hassle, especially at the higher end of the chromatography. The RMX columns seem to be a lot lower. It's a lot cleaner, less noisy. It started going up just a little bit right at the end where all my analytes diluted. This is a blank, or 525 blank, which includes surrogates and internal standards, which is why you see the peaks. But it was reproducible, and from shot to shot, I see the same data. The traditional column had a higher thermal bleed, and while the RMX column stayed low, as you see it. So selectivity, I wanted to be able to make sure that I see what I wanted to see. The two chromatograms, the right and left, is a low-level 525 standard and a mid-level 525 standard. The peaks are all there, sensitivity is there. Again, I see the high thermal bleed on the traditional column towards the end, 20-25 minutes of my run, for both low-level and mid-level standard. And you can even see the RMX column because it's pretty flat. Peak resolution. So one of the things that we need to resolve is benzo K and benzo B fluorantine. This resolution is essential for a few methods. It's a requirement for a few methods, and it's something that most labs struggle with because this is kind of a telltale of when a column needs to be replaced and when a column is going bad. The chromatograms on your left, it's an RMX column. This is when I first put it in, and versus a traditional column. We're getting about 50% resolution versus RMX about 20-25% resolution. But the important thing is, after 6 months of use, I do not see a reason to change this column because my resolution is the same. The resolution is actually pretty much equal to when I put it initially. This is with continuous use, 6 months of use, and with a lot of samples going through it. These are not isomeric peaks, but they're coeluting peaks. Andino 1,2,3 CD-pyrene and dibenzoanthracene will resolve in the RMX column, and traditional column still struggles a little bit. On the bottom end, we have dimethoate With the RMX column, you see more sensitivity for dimethoate, which is sometimes problematic to elute, versus a traditional column. You have a little bit less signal to noise, less sensitivity, less reproducibility. With dimethoate, the peak is there, and it's pretty easy to determine, pretty easy to see. And of course, with linearity, because you have the resolution, that added resolution, added sensitivity, you're able to see things better at the low end. So if you look at the lower end of the calibration, you have better linearity with the RMX columns versus the traditional column. I don't think there's that much linearity also for on the bottom end of the traditional column. That's actually, those peaks are pretty much picking up baseline or some junk in there. But the RMX column, the linearity is there, and I think we could probably go a little bit lower than this, but this pretty much speaks volume. And the recovery. So I have a average recovery of a low-level CCV at 0.1 ppb and a mid-level recovery of CCV. This is for a 525 standard. But what I want to emphasize that this is not a 10 shots and took the average. In terms of linearity, because you have the sensitivity, because you're able to see that low, you could actually get really, really low recoveries, really recoveries that you couldn't see with a traditional column, and you get way more linear calibrations. This is pentachlorophenol, which is a polar compound, sometimes very hard to elute, very hard to stabilize. With RMX column, again, good sensitivity, good linearity. Traditional column, I would be uncomfortable with this peak, and the chromatography and the linearity are missing. And to circle back to the DCM band, because we would need to use less methylene chloride. We're going to have to use less methylene chloride, but we would also need to decrease the volume of the sample mass, or the mass that we're using. Using the column, having the ability to see 20 times lower, 100 times lower, really opens up a lot of opportunities, a lot of things that we could do, and methods that we could analyze at a lower level. The top is a chromatogram of 80 to 70, 20 times lower than what I would need. On the bottom is 100 times lower. This is something super usable. Sensitivity is there. There's no reason we could not run with this type of method. So instead of using, for every method, for every sample for 80 to 70, you utilize about 200 mLs of methylene chloride. Lowering it to by half by using 100 mLs or 50 mLs, it's super attainable using this column. And one of the things that I noticed is the stability of PAHs. So I installed this column like around August of last year of 2025. And that's indicated by the bar in the middle of the graph. The previous columns, the traditional columns, weren't bad. We were getting recoveries of 70 to 110%. It was okay. But once I installed this column, you could see a dramatic change in the stability of PAHs. So if anybody's using or interested in doing PAHs, I strongly recommend this column. It's going to be super stable. And to this date, it's still very stable. So in conclusion, I tested this column through several tests. One, stability, sensitivity, resolution, and efficiency. And in terms of stability, this column was super stable. It's still being stable. It's very reproducible. And the thermal bleed was non-existent. It's not there pretty much. It's not going to affect with your runs. So that it's a green light. In terms of sensitivity, all the peaks of interest were there. My resolution was there. My reporting limit detections were there. I pushed this column a little bit below the limits that I'm looking for, and it produced without any issues. In terms of resolution, I was able to resolve those two isomeric peaks and other non-isomeric peaks efficiently. Not only did I resolve them, I'm able to keep them resolved, right? It's not just where the column is new, but also when the column is 2 months, 3 months, 4 months old, that you know that you have a good quality column. And for that, I give it a thumbs up because the process is still being resolved. In terms of efficiency, the ability for the column to elute all the compounds of interest were there in a timely manner. I'm not running a column for 3 hours or 2 hours. I could produce my full run in 15, 20 minutes efficiently with the right resolution, with the right things that I need. And in terms of robustness, I can't tell you how long this column will last you because I'm still using it. I'm 6.5 months into it or 6 months into it. And I have no indication for me to replace this column. All my resolutions is there, my sensitivity is there. So I can tell you if it's gonna last you 6 months, a year, or 2 years, I don't know, because I'm still using it and there's no reason for me to replace it. And with that, I thank you. And if there's any questions, I'm happy to answer. My emails and the screen. But other than that, I'll pass it on to Ram.
Thank you so much, Eddie, for those amazing presentation and the intense data that you presented. Really appreciate it. Here is RMX resource. If you would like to learn more about the applications discussed here, or if you need additional details about RMX, or you have other applications, we have our website ready with all those resources available. You can download these free of cost and look at the analytes. In addition, if you need additional details, we have our technical support as well as sales support ready to answer your questions. In summary, RMX GC column provides you enhanced detection to hit that sensitivity levels. You'll actually realize less downtime, more discovery in your lab. This column is suitable for a wide variety of analytes, including acid, bases, neutrals, polar compounds, and you'll be able to achieve lower level calibration ranges in terms of picograms on column or ppb levels. And overall, the column would maximize profitability by extending method performance and column lifetime. Thank you very much.
All right, thank you all so much for such a great presentation today. Before we get started on our question and answer session, I would like to remind our audience how to submit questions. You can do so by typing them directly into the Q&A box below the video player. All right, so the first question I have here, Chris, I think might be a really good fit for you to answer, which says, how is the pyridine peak shape on the RMX column and at what concentration is it?
So it's a really good question because first, in order to optimize pyridine peak shape at the front of a chromatogram, you have to make sure that you have a efficient sample transfer to the head of the column. And the way we've done that is we optimize the peak shape for 1,4-dioxane, which means that we have an efficient transfer and we don't have any interference from the methylene chloride. Once we've established that, then we can evaluate the peak shape for the pyridine. And the peak shape on pyridine is outstanding, even at the very low levels. Now, we've run it all the way down to 0.1 picograms on column, and we've calibrated it down to 1 picogram on column. And it certainly is much better than the other columns that we evaluated, not only by peak shape, but by response using MSMS.
Fantastic. We have our next question here, which Eddie, I think might be a fit for you. It says, thank you so much for the deep dive into the analytical data. How do you see this column improve your lab throughput?
So when a column is consistent and with high sensitivity, it cuts down on runtime and troubleshooting, not just runtime troubleshooting, but also conditioning time and just the ability to just run things efficiently. So using a column that can provide provide that type of analysis directly translates into a higher sample throughput and building a more predictable schedule for my staff. It also provides defensible data, right? So if you have peaks, if you have a column that gives you better sensitivity, it's going to give you more defensible data because the peak is going to show a peak and you're not trying to interpret a peak with a low sensitivity. And also cleaner QC performance. If we have QCs that we could barely see the peaks from, having the sensitivity will give you a lot better resolution and a better QC performance, better QC output.
Fantastic. Thank you. We're going to move to our next question here, which Ram Kumar, I think this might be a good fit for you. It's talking about the Integra transfer line. So this person is saying, Integra transfer line is all new to me. Would you be able to explain it in detail?
Excellent, excellent. That's a great point. Yes, Integra transfer line is all new, and we are so glad to bring that to market. So if you look at the Integra transfer line, it's basically a Trimax deactivated tubing without stationary phase. Now, what we are doing is you're putting the transfer line usually at higher temperature, right? And then now, if the transfer line doesn't have stationary phase, that means that there is less gunk that would go onto your mass spec source. That actually reduces the noise level, as well as there is opportunity to improve your signals through this Integra transfer line. Definitely a brand new and a great format for people to try to boost their sensitivity.
All right, fantastic. We'll move to our next question, which Eddie, I think this may also be for you. So putting you on the spot again,
Okay.
would you be able to elaborate more on the benefits you saw with benzo BK and why is it important?
Yeah, so benzo B and benzo K fluorantine, it's a common topic of around the lab. So these two compounds are two pHs isomers that behave pretty much chemically identical. They elute very, very close together, and as you could imagine, it's hard to chromatograph out with a bad column or with an improper column. So because they typically serve as a warning sign when a column needs to be replaced, having a column that resolves these two peaks better for a longer period of time means that we would have decreased the amount of maintenance around the lab or in the instrument. So that means that will boost performances. Also, a lot of the methods do require some sort of resolution between these two. Some are 25%, some are 50%. So if we could maintain that resolution for a longer period of time, allows us to run the system for longer and not have to break the vacuum and change the column more frequent.
Great, thank you. I have a two-part question here for you, Chris. So I'll ask you the first part and then I'll throw you the second one after. This person says they are currently using a 5% phenylalanine phase. How is the selectivity on RMX?
Okay, so the selectivity has been tuned so it's identical to the RXI 5-sil-MS and the DB5-MS. So it's a direct replacement for either one of those. So that selectivity, it's similar to a 5% phenyl, but certainly it's not the same. So it would be different than a pennant phenyl type phase.
Great. And part two of this question is regarding the sensitivity. So in terms of the sensitivity on the column, what level are we talking about?
So the whole idea behind the RMX with the Trimax deactivation is the ability to go much lower and calibrate much lower from nanogram level concentrations all the way down to picogram level concentrations. And this allows those laboratories to running single quad to then run a triple quad, which then translates into extracting less sample and using less methylene chloride for much higher sensitivity, less on column concentration. And that allows a higher throughput and a savings of methylene chloride.
Fantastic. We're going to go to our next question, which I think may be a good fit for both Chris and Eddie to answer. So feel free to jump in, which says, what is the recommended liner for going to lower level detection?
I could jump in if you don't mind, Chris. So I've used a number of liners, and one of the issues with liners is that one of the issues with our methods, at least, is that we have to abide by a NDDT breakdown. If those two don't pass the criteria, then we can't even run any samples, any batches. So when I switched to the Topaz liner, the Topaz line liners, that dramatically and drastically reduced the need for the liner replacement. And the breakdowns were never an issue anymore. So I would recommend the Topaz liner, definitely.
And for a split injection, we recommend the Precision Topaz liner with wool in the center. And in splitless injection, we recommend the wool at the bottom of the inlet. And the reason is the residence time of the compounds in the injection port. The longer a compound spends in the injection port, the greater chance that compound will break down. And if the wool's positioned at the bottom, the temperature is cooler at the bottom. So there's less analyte breakdown for a splitless hold, which would actually hold those compounds in the inlet for a longer period of time than a split. Whereas with a split, you have a 10 times faster transfer of the sample onto the column, assuming it's a 10-to-1 split. And therefore, we want the wool in the middle to get rapid vaporization and rapid sample transfer to the head of the column.
All right, great. Thank you both for tackling that one. We'll move to our next question, which again, Eddie, I think might be for you, which says, what are the different matrices you have looked at on RMX and how is the performance?
So we ran from drinking water to wastewater, drinking water, surface, and wastewater. And it was quite a bit of it. It was quite a lot of samples that went through those columns, through the column. And I don't think I saw any weird issues with wastewater. Looking at wastewater, in retrospect, looking at wastewater analysis and drinking water analysis, both look the same, regardless of what was being injected. So it's hard for me to say how much of what, because we were just running samples. A lot of things come in with different types of matrices, and we just didn't keep track of what matrix we just running the samples. But from our perspective, we couldn't tell whether we shot at drinking water or a wastewater, which speaks volume of the type of chromatography and the column itself.
All right, fantastic. With that, we will wrap up today's event. Any questions that weren't answered during the session will be covered offline via email. I want to thank the audience so much for attending and for participating in today's event, especially with the polling questions. I would also like to thank our speakers very much for their time and knowledge today, and our sponsor, ResTech, for making today's webcast possible. We would like to ask everyone in the audience to participate in a brief survey. This survey will appear on your screen after today's presentation has ended. You receive an email alerting you, and this webcast will be available for replay, and we invite you to forward that to any colleagues who may have missed today's webinar. Thank you everyone for joining, and we'll see you next time. Have a great rest of your day.
Thank you.
All right, great, thanks.